Okay, today's podcast is going to cover the cell cycle. Of course, we all know that we humans are multicellular organisms. We have trillions of cells, but we started off as one cell when a sperm cell fertilized an egg cell, and we became that first cell known as a zygote. But that zygote went through some cell division. It divided into 2 cells, and those divided into 4 cells, and then those divided into 8 cells, and so on. And now we are trillions of cells. But of course, when we stop growing, that doesn't mean that our cells stop going through the cell cycle. There's lots of different reasons that cells need to still continue to go through the cell cycle and then eventually divide. One example I can think of right now is red blood cells. So your red blood cells, they live to be about 120 days, and they wear out just like any cell, and they need to be replaced. And the cell cycle offers a way that those cells can be replaced by new cells. For unicellular organisms, the cell cycle or the cell division causes a new cell to be created. And so that's just a form of reproduction for unicellular organisms. But as we talk about the cell cycle, I need you to understand that the cell cycle in mitosis is not just as simple as a cell dividing in half. You guys are going to learn about the different steps involved with this process known as the cell cycle. These steps are specific in the certain tasks or jobs that are taking, or the processes that are taking place during the cell cycle, but the cell cycle isn't the same amount of time for every single cell. Of course, some cells, they might take a few hours to go through the cell cycle. Some cells might only take 20 minutes to go through the entire cell cycle. And then some might take longer or shorter than that. Most of the life cycle of a eukaryotic cell is spent in a period of growth and development called interphase. Interphase is labeled as the letter I when you look at the cell cycle. Cells that divide, they'll spend about 95% of their time in interphase. Cells in your body that no longer divide, such as nerve or muscle cells, those are always in interphase. But an actively dividing cell, such as skin cell, copies its genetic material and prepares for cell division during this phase. So you might be asking, why is it important for a cell to copy its genetic material? Well, I like to use an analogy, maybe a football team, for example. Football players, the coach is going to give them a copy of the playbook. And so every player needs a copy of all the different plays that are going to be on that team. Okay, if a new player comes to join the team midway through the season or the next season, they're going to need a copy of that playbook so that they can learn the plays and carry out all the jobs that the coach wants. So in the same way, a cell needs to make copies of its DNA, which is the code of life, so that each cell that's made afterwards will still contain the playbook. So this genetic code has been passed on from generations to generations. So the same DNA that your grandparents had has been being passed down to the offspring and passed down to that generation until now you have some of the same exact DNA that all your grandparents had. Now when you have a new cell divide, okay, it needs to have the same genetic code or the same DNA as the mother cell. And so that's really important to have DNA copy itself in the cell cycle. And this takes place in interphase. So even though interphase is sometimes referred to as the resting stage, there's still lots of stuff going on. This interphase is subcategorized into some other phases. We have the gap 0 phase, that's the resting phase. But cells will go through a gap 1 phase. That's when cells are going to increase in size, so they're getting a little bit bigger. Then there is the S phase. The S phase is where the DNA is copying itself. Sometimes we refer to that as DNA replication. But then after that, there is the gap 2 phase, or the G2 phase. And then that's where the cell is going to continue to increase in size. So it's preparing itself to go through this division. So after the cell has went through the entire interphase, it's now copied its DNA, it's prepared to divide. Okay, we call this next phase, the mitotic phase, or the M phase. This is where the cell is going to actually separate the two copies of DNA and complete the division process to form two complete new cells. So now I want to talk a little bit about mitosis specifically, this stage where the cell is going to divide. We're going to go over the phases or the steps in mitosis, and then we'll wrap it up. How we're going to remember the stages of mitosis is we're going to remember the letters P, M, A, T, PMAT. That's going to stand for the different steps: prophase, metaphase, anaphase, and telophase. We should talk a little bit about chromosomes. A chromosome is a structure in the nucleus that contains the genetic material for the cell. So basically, a chromosome are these histones, these special proteins, and it has DNA wrapped around them. When the cell gets ready to divide, all these duplicated chromosomes, they're going to coil tightly into thickened, identical strands called chromatids. During mitosis, the first phase is prophase, and how we're going to know that we're in prophase is you're going to be able to see these pairs of chromatids. They're going to be fully visible because they've coiled up and they've tightly coiled up and thickened, and so now we can see them. The nucleolus and the nuclear membrane are going to disappear during prophase, and the two small structures called centrioles are going to move to opposite ends of the cell. Between the centrioles, thread-like spindle fibers are going to begin to stretch across the cell. And plant cells, they also form a spindle fiber during mitosis, but they don't have centrioles. Now we're ready for the second step of mitosis, which is known as metaphase. Metaphase is really easy to identify because you're going to see the chromatids line up across the center of the cell. And so when we're looking through the microscope, you'll be able to identify metaphase really easily just because those chromatids are lined up in the middle. Now, as the chromosomes or chromatids line up in the middle, the spindle fibers are going to attach to each side of the chromatids. Now we're going to move into anaphase. And so those spindle fibers are attached to the chromatids, and they're attached at the centromere. And the centromeres are going to divide as the spindle fibers shorten. Each pair of chromatids are going to separate, and the chromatids are going to begin to move to the opposite ends of the cell. And the cells are the separated chromosomes are now they're called chromosomes. In the final step of mitosis, known as telophase or telophase, the spindle fibers are going to start to disappear, the chromosomes are going to start to uncoil, and now the new nucleus is going to form. For most cells, after the nucleus has divided, the cytoplasm separates and two new cells are formed. In animal cells, the cell membrane pinches in the middle like a balloon with a string tightened around it, and the cytoplasm divides. But in plants, the cell plate is going to form, and new cell walls are going to form along the cell plate, and a new cell membrane is going to develop inside the cell walls. And following the division of the cytoplasm, most new cells begin the period of growth known as interphase all over again. So that was just an overview of what the entire cell cycle is. It starts off, it spends most of its life in interphase, and then it's going to go through the mitotic phase where we have each of those steps. I hope you guys learned a little bit. We're going to be looking at some slides to identify some of these stages, and thank you very much.